conference again. We hope everybody is well, and we also hope that you are enjoying our meeting. And we are really delighted to welcome you today for the session number 19. It's a highly classified session, let's say. It's named as other topics. So I am supposed to make a very short presentation of your talks. We try to do it as fast as possible. I probably need 9-10 minutes to finish this presentation, and I'm going to be sorry in advance because I mean I read all abstracts, I watched all the presentations, but there might be some things that I misunderstand. So I'm sorry in advance for that. I will project my screen now for you. Just a second. Today we have 9 presentations, 9 talks, let's say, that we are going to be discussing. And well, I put in the order that you probably have in the Excel file. But today, like I told you, we have 9 presentations. So we will start. Let me start here with, sorry for the full screen. Everybody seeing and listening well? Can you hear me well? Is everything all right?
Yes.
Okay, so let's go. So, like I told you, we have 9 presentations, 9 talks that we are supposed to discuss today. And well, the first talk is just because my screen is at the top of the PowerPoint presentation, so I cannot see well. So we'll minimize, and I think it's going to be easier. Well, the first talk is led by Whitley. I'm sorry about the names. I cannot pronounce well your names. But anyway, the title of the talk is Landscape and Freshwater Biodiversity Conservation in the Anthropocene, the Contribution of Artificial Water Bodies. So this, I mean, I decided to only put the goals and also the hypothesis of each one of your works in this presentation. So it's going to be very straightforward. I'll try to be as fast as possible with your presentation. So the main questions of this work was, can artificial water bodies bring a contribution to freshwater biodiversity conservation? So the authors investigated this question. They used 291 ponds. It's a very interesting work. And these artificial water bodies were distributed along Switzerland, France, and Spain. And they compared a selection of these artificial water bodies with natural ponds. I mean, the main results that they found is that the artificial water bodies host, in average, only 50% of the regional species pool that were sampled in these lakes. Fish ponds contribution was highest, and mountain watering pond lowest concerning the biodiversity contribution. They forecasted the future human-dominated landscapes will be particularly impoverished in freshwater biodiversity. And the second talk is entitled as Land Cover is the main correlate of phytoplankton biodiversity in subtropical coastal shallow lakes. And the first author of this work is Ana Paula Tavares Costa. This work is already published. And the main goal of this work was to understand how limnological heterogeneity, primary productivity, spatial extent, connectivity among lakes could actually be related with within lake biodiversity of phytoplankton in 25 lakes distributed in the southern part of Brazil. And they found some interesting correlations between these environmental variables and biodiversity. They used different dissimilarity indices for this analysis. And biodiversity was driven mostly by environmental heterogeneity and wetlands in these systems. And also biodiversity, as I told you, was strongly correlated with wetlands. I mean, there was an increase in biodiversity with connection with wetlands. And also interesting that the biodiversity also depends, I mean, the response of the biodiversity of phytoplankton also depends on the kind of dissimilarity coefficients that are being used. So it's necessary to understand these two dissimilarity coefficients to make a better prediction of change in the phytoplankton community structure. And the paper is already published as well. The third work is the effects of multiple stressors on invertebrate communities in shallow freshwater habitats. The first author is Nora Kiefeller. I'm sorry about the pronunciation. And the main goal of this work was to understand how different environmental stressors like pesticides and nitrates can have an influence on the food web dynamics in these shallow lakes. The authors analyzed how these combined stressors may have an influence on the dynamics and functional diversity and abundances of zooplankton in lakes. They performed some experiments to understand that. The fourth work title is Stressor Interactions in Shallow Lakes Aquatic Ecosystems, a microcosm study to better understand regime shifts under multiple stress. The first author is Maxfield Schmidt-Jensen. And the main aim of this work was to disentangle how these environmental stressors, like in the former work, and in global warming, could potentially be related or occurring in two alternative stable states in lakes, in ponds, actually. So here there is some summary hypothesis. The first hypothesis was that agricultural stressors like nutrients and pesticides could cause multi-directional or contrasting effects and therefore result in antagonistic interaction types. I will not talk about the results that were found, but we have time to discuss that. The second hypothesis was that combined stressors could have a regime shift between alternative stable states. And the third and final hypothesis was that these combined stressors modulate the thresholds for regime shifts in these ponds. There are some conclusions here that the agricultural stressors result in regime shifts when applied in combination. The shifts are mediated by enhanced growth of phytoplankton. There are some also indirect effects mediated by phytoplankton, just in the presence of pesticides. There were some regime shifts in shallow lakes, and these regime shifts in shallow lakes are actually enhanced by global warming. And the fifth work of today is entitled as Diversity of Testate Amoeba associated to different aquatic environments. First ecological study and first step to future research as bioindicators in the Ibera system. And the first author of this work is, I think it's wrong here. Well, here is Montliareco, I'm not sure. You can correct me afterwards. Well, the study goals was to use this test state amoeba as environmental indicators to aquatic monitoring due to their rapid response to stressful environmental changes, and also to evaluate the composition and abundances of test state amoeba communities among different sites in Iberia. And they use anthropized and non-anthropized environments, and also different seasons to understand how these differences in the systems and also differences in the seasons could have an influence on the composition and abundance of a test state amoeba. So here, some results just showing that there is strong differences when you consider anthropized and non-anthropized environments. And this study confirms for the first time the presence of the 14 species of test state amoeba in the Iberia system. The study of test state amoeba communities is a key step to use them as a bioindicators on water bodies, according to the authors. And the sixth talk of today is entitled as Linking Macroecological Patterns to Microecological Dynamics of Community Assembly in Meta Communities. This work is led by Friedrich Engel, and there is a background, of course, behind this work. I'm not going to tell you the details, but there are some disagreements, let's say, between the patterns between, there is a disagreement with natural studies and experiment studies concerning the relationship between chlorophyll and zooplankton, for example, and this work tries to somehow reconcile this this lack of it's not lack of, well, we'll try to explain the hypothesis. I'm sorry, I'm a little bit nervous now, but I will try to do my best to explain this again. So there is some, in this work, there is a disagreement, let's say, between the patterns that is found in nature and experiments. Short-term experiments usually ignore the effects of colonization and extinction dynamics that may somehow have an influence on the community structure. Something that you can investigate better when you use the macro scale perspective. That's the general idea of the work. And try to connect base macroecological patterns and microecological dynamics of community assembly in meta communities. And they use some emerging statistical methods to understand these relationships. And the seventh talk of today is entitled as Disentangling the Role of Potential Control Factors of Phytoplankton Productivity in Subtropical Dystrophic Shallow Lakes in Uruguay. And this work is, the first author of this work is Baladan. Well, the main study goals is try to understand how different factors like light, colored, dissolved organic matter and nutrients could have an influence on the phytoplankton productivity in a dystrophic lake like Laguna del Cisne in Uruguay. So Baladan performed some experiments. It's a factorial design experiment to try to understand how these factors could have an influence on the phytoplankton productivity in these lakes, in this lake. They also discuss the importance of light to increase or decrease phytoplankton productivity in these lakes, and also discuss the threats of the increase, let's say, of nutrients and also decrease of light irradiance in these lakes could have an impact on the water quality and consequently other ecosystem function in these trophic lakes. And these results are important for management decisions. The 8th talk today is entitled as Black and White, Day and Night, Fish Diurnal Movements in 2 different Amazonian lakes. The first author of this work is Prit Zingel, from the Estonian University in Europe. So the main study goals was to understand the impact of changing dissolved oxygen concentrations in the fish movements in white and dark lakes in the Amazon in Brazil. They also compared high and low water seasons, compared the fish catches during the high and low water seasons, and then divided their sampling in the littoral and the pelagic zones. And more, actually, sorry, in more discussions, we have the opportunity to discuss this work now, today, according to the authors. And the 8th talk today is entitled as Phytoplankton phagotrophy by cryptophyte and chrysophyte across nutrients and light gradients using different measurements techniques. The first author of this work is Mariana, and she's here also to discuss with us the results of her work. And here, just a small background about how environmental change may benefit mixotrophic activity in lakes. And within this context, Mariana tries to evaluate, actually through the experiments, phagotrophic activity of these 2 groups of phytoplankton, cryptophytes and chrysophytes, using 3 different approaches: flow cytometry, efflorescence microscope, and LISO tracker. And she will discuss a little bit about this when asked here in our discussion. So the main conclusions that she had is that the cryptophyte, the nutrients is the main resource for cryptophyte and light for chrysophyte. Flow cytometry showed a high capacity to distinguish grazing rates, and LISO tracker can be used as a quantitative, qualitative method. However, cannot be used to quantify photographic activity. And just to remind you that this session is being recorded. So I decided to give you 90 minutes to discuss each one of the talks because our time is short and there are a lot of talks to be discussed here. And I'm very sorry for the presentation that I did of your work. I'm pretty sure that you can do it much better than I can. I will finish the presentation here. And unfortunately, there are no questions in the Moodle platform. So I will keep the order that sorry, just a second. I will keep the order that the organizers have already decided before the conference. So I think we can start our discussion with the work by Beat Ortoli. I'm sorry about the pronunciation of your name.
No problem, no problem.
But we are open for questions. I would like to ask you to write your questions and comments in the chat area if you want, just to keep it organized. Anyone who wants to ask questions, people from that they are not actually presenting, they are also welcome to ask questions to the presenters today. You're free to ask questions with the microphone as well, no problem. So any questions for the first work? Just to remember is that landscape and freshwater biodiversity conservation, the Anthropocene, the contribution of artificial water bodies. That's the work that we are supposed to discuss now. I think Matilde has the first question.
Yeah, first of all, I want to thank Beat Ortoli for this very nice presentation. It was very interesting. Your final recommendations were to enhance the structure of artificial systems to increase biodiversity, because you discovered this gap in diversity between artificial and natural ponds. I have two questions. The first one, did you correlate structural features with biodiversity to support this finding? And second, what plays the age of the systems a role? Are they still in a establishment phase, or increases biodiversity over time?
Yes, good question. Yes, and first, before answering, I want to thank you, Rosenberg, for doing this nice work. It was nice to make this different, abstract, very different talk. Okay, thank you. For coming back to my presentation, yes, we did analyze. It's clear that we didn't show all this now. And for some type of pond, we made some special investigation to know which environmental factor can enhance the biodiversity. For example, we did it in especially for urban ponds. We did not do it for every type of artificial ponds, but we did it, for example, of urban pond, and it was a clear evidence that this was one of the main factors. This is the presence of vegetation on the shoreline. This was and this is, for example, very easy to do it. We can do it in alpine water bodies. We can do it in the urban water bodies. It's a simple measure. And even we know, for example, that they develop new system of floating islands. This could be another possibility. It's to enhance the presence of emergent vegetation on the side, but it will also be in the middle of the ponds with floating islands. This is very interesting because they are insensitive to the variation of water level. That means that in artificial systems, they are still floating. I know that in France, they are developing them a lot. It's more for production of fishes, but it's also very good for the other type of biodiversity. So this was to answer to your first question. We have extended data also about the other variable, but this was one of the main one. The second question is about age. We try to avoid this problem by choosing, I think, systems that were relatively young, but that means that I have 10 years. So 10 years is very different, for example, that in natural systems that are 4,000 years old. And this has also an influence. We did evaluate the colonization rate, for example, in systems that we built, a new system. And the main colonization pattern occur in the 10 first years, and then you have still an increase until 50 years. So it will be good perhaps to also do such analysis with older ponds, but for example, with gravel pit ponds, it's not possible. Gravel pit ponds are all young, and but when you let them evolve, after they have also another type of state. But to come back to your question, age is an important factor, and also in pioneer state, you have also pioneer species, especially for amphibian. Amphibian, you will not find the same species in natural ponds, for example, and in artificial gravel pit ponds that are 5 year or 6 year old. And this is also an important topic that we didn't present here, but you can sometimes have some originality, and it's why in the last analysis that I presented, the nested net analysis, you see that the gravel pit ponds are a little bit separated from the natural ponds. It's because they are often pioneer system with pioneer species, and these pioneer species, you find them nearly only in such type of pond. As the natural processes of creating pond today do not more exist, or I mean, they exist still, but they are relictual. So some artificial systems that are built can bring a contribution also on this side, and but we didn't present this in the this investigation. Yeah.
Okay. Any more
Does
questions?
it answer? Okay. Yeah, okay.
No questions?
I have one question. So despite the age of some of them, what are the main environmental differences between the artificial and the natural ponds that could lead to this different species composition?
You mean in the relation with age?
Yeah, or I mean, I know that age would also affect the environment, but what I meant more, what kind of environmental factors, did you measure any of the nutrient levels and stuff like that in the
yes, okay. So I think really that it was the diversity of habitats that was the main difference. The diversity of habitats, and like you saw perhaps in the first picture, sometimes you had also an artificial substrate with a plastic sheet or something like this. And it's not in favor of a growing macrophyte. And in consequence, you don't have large macrophyte belts. And so, and this was really the main difference. You in the chemistry, water chemistry, not really, because it depended really of the watershed. Even natural pond can be in a really impacted watersheds, and so, and it was not the difference. Even in some artificial pond, you could have a nice quality of water linked from to rainfall and so on, and it was not a characteristic of artificial ponds to have a less good water quality. Yeah.
Does anybody have any questions? I have one. I was wondering about the community structures within each one of these groups of ponds that you sampled. Do you have any idea about the effects of non-native species, for example? I don't know if you are writing anything else about the species that are not native from these systems.
Yes, we had non-native species, but in every type of water body, in artificial and natural. I mean, it's more often the presence of the man, of human presence that is linked to this species. So when you are near of a city, even if the pond is natural, you will have some exotic fishes or exotic plant. But perhaps the main difference is in urban ponds. Urban ponds are really dominated by exotic species, especially for the macrophyte, you know, because people plant them. And this was the main difference that we saw, is that the species richness of macrophyte was good in urban area, but only because people plant them. And when they plant them, they have not the good knowledge. It's only because of they don't have knowledge. They buy what they find, and sometimes they buy some species that are invasive, and we find them a lot. For example, Elodea canadensis and Elodea nutali are two species considered as invasive. You find them nearly everywhere, but also in natural ponds now. Yeah.
Okay, thank you. I think we have to move on to the next talk.
Okay, yes, thank you for the question. Yeah.
Thank you very much, Beat. I don't know if I pronounced your name again
correctly again. But no problem.
But okay, let's move on to the next talk. The next talk is entitled as Land Cover is the main correlate of phytoplankton beta diversity in subtropical coastal shallow lakes by Ana Paula Tavares Costa. Are you there, Ana Paula?
Hi, everyone. Good morning.
Is it okay if you turn your camera on, or if it's fine? I don't know. I see if your camera is working properly.
Yes.
Does anybody have any questions? Feel free to ask Anna. Nobody has questions?
No.
I guess I have a question, but it's very general, and I don't know if you know this because it's maybe statistical. But so you use all these different indices, but I know everything takes time. So if you had to choose one that you would think would be the most reliable, would you make a recommendation which one to use?
You can write the question, please. So I write the answer in the chat. My English still is not very good to speak and listening. Sorry.
It's okay, but we understand you well, actually. Okay, Anna, can you see Frederick question? I think she's not here anymore. Well, I think we should move on then to the next talk to not waste much time. But Anna, you are welcome to answer Frederick questions at the end of the discussion, because I mean we are short on time, so unfortunately we cannot wait for you. I don't know if you had any problem with your internet or anyway. Let's move to the next talk. The next is entitled as the effects of multiple stressors on invertebrate communities in shallow freshwater habitats by Nora Kipferle. So does anybody have any question? No questions?
Anna answered at the chat.
Oh, okay. I think everybody can read that. Actually, it's a very awkward situation now. I don't know actually how to proceed. If you could help me with any suggestion.
Maybe at the end of the meeting, Anna can answer that.
Yeah, I think it's probably the best idea. I don't know.
Yeah. Because she's nervous about the English, so she wrote.
I think it was also more, I thought if we maybe have time in the end for a little bit of discussing different things, because usually in my experiments I always used Bray Curtis, but I'm very curious of why other people use other methods and stuff like that. So that's why I asked, but it's not super important, so maybe we can move on to the next talk.
Okay, I'm sorry, Nora, for making you wait a bit, but does anybody have any questions about Nora's work?
Can you repeat the title?
Yeah, sure. Effects of multiple stressors on invertebrate communities in shallow freshwater habitats.
Well, if nobody has a question, I'm quartering the talk. Anyhow, I have a question to Nora, perhaps to start the discussion. You also referred to stress interactions in terms of synergistics and antagonistics. Could you quickly explain how you came to these conclusions and what does this mean for your output, or how do you interpret these interaction types? What's the consequences of these?
To analyze for the interactions between the stressors, we used a negative binomial model with the program SPSS. And then this gave us a predicted mean how the population should have reacted to this combination of stressors, which was extrapolated from the single stressor reactions. And if our result in the experiment diverged significantly from this predicted mean, then we had a significant interaction effect, either antagonistic or synergistic. And just let me check. We had quite a lot of antagonistic interaction effects for the zooplankton. So often the effect of the combined stressors was not as devastating to the population as you would have thought, maybe, because it was not additive, it was less than additive. And when I look at my notes, only the copepods in one experiment showed synergistic effects. Yes, but really most of the time we had antagonistic effects, meaning that two stressors did not interact additive, but less than additive. Which is good news,
sorry.
I guess. Yes, please speak.
Sorry. Actually, I have a curiosity. You performed 2 experiments, and in the mesocosm experiments, you basically didn't have any kind of effect. Could you say something? What happened with the mesocosm?
So the mesocosm was a big cooperation project. So 2 of my colleagues are sitting here, Mechtel Schmidt-Jansen and Elisabeth Groß. And we did analyze lots of organisms in these mesocosms. So we also did a tight observation of the macrophytes and the periphyton, the phytoplankton. We had snails in there, also mussels, and we checked the water parameters regularly. And we did have significant effects, but sadly not for my part, which was the zooplankton, which is one of the reasons why I did additional microcosms after the mesocosms, because to get some results and to try to interpret those results. And if we look at the data, we can see that something happened, but the way the mesocosm experiment was designed is not, in hindsight, is not ideal for analyzing zooplankton communities. We have not enough samples and not enough replicates to really have a statistical firm grip of what happened to those highly oscillating zooplankton populations over time.
I have just one more question about it, but it's about the species composition. Can you actually say that changes in species composition are actually driven by the stressors, or it's just a successional change, let's say, driven by competition among species? I was just wondering that.
So
or maybe
of course,
I'm totally wrong.
of course, we had a general change in species because of our experimental system. So the animals, the species composition we put in at the start of the experiment was completely different than the one we had in the end. But in general, the species that seemed to be fit for our experimental system, they were kind of the same in all of our 16 enclosures. And then there we had significant differences in the composition of this final community in between the treatments. So we had an effect of our toxic mixture of our treatment there.
Okay, thank you very much. Does anybody have any questions? Yes, feel free to ask a bit.
Yes, I was wondering about the ARO, is a runoff, ARO that you use. I mean, you can have thousands of what I call some cocktails, you know, and you have chosen one. And I want to know, how did you choose it? And is it chosen as a representative of what you could find elsewhere on the...
So Mechthild needs to correct me if I'm if I'm citing this wrong. We wanted to choose a mixture. One is a pesticide, one is a herbicide, and one is a fungicide. That was important to us to have this whole array.
Okay.
And also the copper, which is a metal that is, for example, used also in organic culture. And then we chose the 3 organic toxins. We chose ones that are still used often. They are still in use. And yes.
Okay. So,
maybe Mechthild wants to add something.
Yeah, it's perfectly described. The only point I can add is that we also chose the toxicants according to their occurrence in the environment. For example, the herbicide we found in over 80% of samples we 2 years ago sampled in small streams in Germany. So this is really a toxicant which is present in Germany, probably also in Switzerland, in France, and across Europe. So there is some, on one hand, they are representative for a group of modes of actions, and on the other hand, they are present in the environment. This was a rationale for choosing.
Okay, yes, thank you.
Okay, does anybody have any other questions? Actually, we can take the opportunity to start discussing Mechthild work. I don't know if my pronunciation was correct. I tried to imitate Nora now, but I think, well, since it's basically the same project, we can continue discussing, but of course, related to Mechthild work. Does anybody have any question? No questions? I have one. It's about the combined effect of stressors related to the alternative stable states. Actually, it's I was just wondering if these alternative stable states that you claimed in your work could be generalized, for example, like Beat asked a few minutes ago, because there are so many different types of pesticides, for example, that can be used in the agriculture fields, especially here in Brazil, for example. I was just wondering if the same pattern could be generalized when you say that these shifts in the regimes would occur in these experiments that you performed.
Yeah, that's actually a very good question. Yes, I think it could be generalized because the mechanisms we found behind these shifts refer to mechanisms we also find for other environmental variables. For example, we found an enhancement of growth, which was in our study related to nitrogen, but in literature is also reported to phosphorus. That's the theory coming from Scheffer, from the original observations. So I think all variables enhancing phytoplankton growth are on risk to cause this. And on the other hand, all mechanisms related to that, which was the shift between the phytoplankton dominance to the macrophyte dominance to the phytoplankton dominance. Interestingly, the stressors which we expected to inhibit the growth didn't show high effects, which was probably coming from the low doses we chose, but they enhanced the effects in the other stressor combinations, which we conclude as something from nothing. So it means that we could generalize that toxicants which refer to these modes of actions, for example, the pesticides we apply on our streams, might be prone to cause these kinds of effects. So we could generalize to general modes of actions of in this case, the pesticides, the herbicides we put out. I expect that the same output if we use all the other pesticides which share the modes of action. Yeah. I hope this is answering your question.
Yeah, you did. Thank you very much. Does anybody have any other questions to Mertil?
I've more an addition, if I can. So
yeah, sure.
I'm also part of this project, and we are spreading a little bit. So there are more talks in the S10 session on Thursday and Friday. And what is interesting is that we observed in our experiments, so we have a range of microcosm and mesocosm experiments. We also have a lot of indirect effects and some effects that are canceling each other. So it's a very complex system. And what was also interesting is to see that if you use only primary producers and you use the first trophic consumer level, then things can also change again. So in some studies, we have then stronger effects when the consumers are included, specifically benthic consumers. So it's I think that there are not enough studies yet on this pesticide nutrient interactions, and we've started this quite some time also, and very often you see that the nutrients are overruling, but we don't think that it is always the case.
Okay, thank you for your comment, Elizabeth. Does anybody have any other comments or questions? Otherwise, we should move on to the next talk. Okay, so let's move on to the next talk. The next talk is the diversity of testate amoeba associated to different aquatic environments. First ecological study and first step to future research as bioindicators in the Ibera system, by Florencia Areco. Are you there, Florencia? Well, I think she didn't show up, so we should move on to the next one then. It's by Frederick. I'm sorry for the pronunciation, Frederick, I don't know, Engel, linking macroecological patterns to microecological dynamics of community assembly in meta-communities. I'm sorry for my presentation of your work. I know it was awful, but we now have the opportunity to discuss a little bit about your work.
It's okay. You close also with the pronunciation. It's Friederike. But I understand. I don't know, I wanted to say this. I'm also more of an experimental ecologist. So my whole project that I presented here was a little bit also for me out of my comfort zone. And so I understand that for a lot of people, it was probably a little bit like, ooh, a lot of different methods and stuff like that. So don't worry, I thought your presentation was fine. I mean, if I had to present that, probably I would have done much worse. So don't worry.
Being nice, but thanks. But well, anyway, does anybody have any question? Okay, I have one. Let's see if it makes sense. Tell me if it's totally nonsense.
Mm-hmm.
In your presentation, you were trying to somehow reconcile, try to understand actually the patterns that you usually see in a smaller scale, like in experiments, like you said, in a macroecological scale.
Mm-hmm.
I was just wondering if within the models that you were trying to build, you can include, for example, speciation and drift. I don't know if I saw it in your presentation, but I remember that you added selection. If I'm not wrong, you also added dispersion, but I can't remember right now. But can you actually include these four, let's say, processes that are so discussed now in the community ecology? That I mean, these processes was highlighted by Valand in 2009, if I'm not wrong. But can you include these four processes, let's say dispersal, selection, speciation, within these models that you are trying to build?
Yeah, so the short answer is yes, but only indirectly, because, for example, the joint species distribution model, it's a latent variable approach, so it only relates, you kind of assume things that you can't directly observe, but it's supposed to observe those things because you have the systems that you look at have been there for a long time. So all of these processes can have taken place because the time is there, compared to experiments where the time is simply not enough, and a lot of times the scale is not enough to have these processes happening. And so the problem is that, no, we cannot directly say, oh, this is caused by speciation or this is caused by dispersal, but you can relate it to the processes and see what is most likely happening, especially if you include then the distance between the lakes or the flow connectivity between the lakes, because then you can see how are they related with each other, what species are in what lake, and yeah, how long it theoretically would have taken for a species to speciate in a new environment, if it is found only in one environment, for example. So I guess the short answer is not 100%, but you can infer at least some of these things from the methods used. But I think that's why it's still important to combine always with experiments, because you just, if you want to see real processes and patterns, you have to know the system really well and observe it over a time, so you see what's actually happening.
I was just wondering, I mean, this pattern that you saw relating chlorophyll with zooplankton, phytoplankton, zooplankton anyway, in the experiment, you generally find in the field a very strong relationship between zooplankton and phytoplankton, like you said, and in the experiments you can find positive, negative, or no effect. Why does it take place in the experiment? Why is so messy, let's say? I would expect the opposite, actually, because you simplify the environment. It's a microcosm, a mesocosm experiment, so probably the response would be more straightforward, let's say. Right,
but I think the main reason why you see these differences is that depending on the system, in experiments you have very clear definitions of trophic levels. For example, you might add a few, like a community of phytoplankton and a community of zooplankton, but then some experiments might also add a community of fish as predators, and so that changes the whole dynamics of the system. So depending on how the experiment is set up, that can really change how the dynamics of the of the whole community with all trophic levels works. But in nature, you have so many little levels, I would say, that in experiments usually are not accounted for. And so so to see a really clear pattern in nature is difficult, because even when you take really good samples of, try to get every organism in the lake, you still don't really know all of the environmental factors and all of those things. And so I think why the patterns in experiments are variable and also extreme is that you really control for what you put in, and so also you pretty much control what you can get out, because you're excluding a lot of the small the smallest changes that could happen in nature.
Okay, thank you. Does anybody have any questions?
Yeah.
Please feel free, Elizabeth, to talk.
Yeah, it's very interesting that you're also using SEM models, because, well, we are kind of fond of them also for some of our results. And it's interesting that you have more the effects of the environment. So I think it's similar to what you explained just now, that it's the complexity or the variability of the system that is explaining mostly of the chlorophyll development. Am I getting it right?
Yes, that's definitely right at this point. And the problem with this is too, in the model, we started out with a really complex model, and then it became too complicated to actually interpret. And so that's when we pared it down. But now I again feel like maybe we're missing something important that we didn't put in. But yeah.
Okay. And just one more question for the zooplankton. You have the biomass, but do you also have the size? Because this is a crucial factor also on the control of increasing.
Yes, we do have information on the size, but I haven't used it yet. So maybe that's a really good point, because that could definitely affect the relationship with phytoplankton. We thought actually about splitting them up into size classes, also for the phytoplankton, to see what's edible and what's not. So that's a good point that maybe I should look at more detail. Yeah, thank you.
Any further questions? Okay, thank you, Frederica. Probably better now, the pronunciation. Thank you very much for your discussion, and your work was very interesting. I'm looking forward to seeing future publications of your work as well. So let's move on to the next talk. It's a work that was led by Claudia Baladan from Uruguay, and the title is Disentangling the Role of Potential Control Factors of Phytoplankton Productivity in Subtropical Dystrophic Shallow Lakes in Laguna del Cisne, Uruguay. So does anybody have any questions to Claudia?
Hello, good morning to everyone.
Okay, go ahead, Bit.
Okay, just a small question of understanding. You have a lot of dissolved organic matter that give a color to the water. From where is coming this organic matter? Is that trees or what is?
No, that's a very interesting question. That's the most interesting about this lake. The CDOM comes from a wetland that is next to the lake, and it covers the 25% of the basin area, and it is the source of the CDOM, because we have also seen a gradient in the lake, in the area of the lake, that next to the wetland source, you can see that the color of the water is more brown than when you are far away from that area. So the CDOMs comes from there, and we believe that it is important to recognize that, as this is an important area to preserve, because it is giving the color to the lake. And we believe that we found that in the lake we have a low phytoplankton productivity, and we believe that it is that is due to the color of the lake, of the water, due to the low transparency. And we believe that that wetland is an area, a very important area.
Okay, thank you. And it is also the source of the nutrients? The wetland is a source of the color and also of the nutrients, or?
Well, in this area, there are many activities. We have forestation, we have some agriculture activities, and we believe that the source of nutrients are coming from anthropogenic causes that comes from all these activities associated to the humans. And we believe that the wetland is providing it's called organic matter, but all the nutrients are coming from these activities. In fact, we have a graph that I presented in the presentation, that we found that we correlate the increase of the nutrient level. We correlated to the use of the soil during the years, and we found that as the use of the soil was increasing, the nutrient level was also increasing. And then there was a decay. The use of the soil decreased, and so decreased the nutrient levels. This indeed needs further analysis, but we can see that it's a tendency between those two variables.
Okay, thank you for the information.
Thank you.
Any further questions? Well, I have one. I was actually wondering what is low and high concentration of nutrients? Could you say something about that?
Well, we found that the highest level of phosphorus concentration was during the year 2012, and it was about 700 micrograms per liter. Yes, and that is really high, a really high concentration of phosphorus for a lake that it is important to take into account that this lake serves as a source of potable water. And this level of 700 micrograms per liter is really above the permitted level for a source of potable water according to our laws. So that I consider that is a really high level. And we found that despite that, we don't have any phytoplankton blooms. So something is happening there, that despite having this high level of nutrients, phytoplankton is not developing as it happens in other lakes. For example, other lakes in Uruguay with lower nutrient concentration, lower than 700, are developing cyanobacterial blooms. But in this lake, it doesn't happen that. It has happened in one occasion during a summer, but it's not common. So we wanted to investigate why we have this discrepancy between the phytoplankton productivity expressed and the expected. the phytoplankton productivity expected.
Okay, but well, do you have an idea about the phytoplankton community structure? Which species is dominant? Can you say something about that, or you don't have this information?
No, this lake, there aren't many studies in that case. We have an institution, a governmental institution that studies the composition of the phytoplankton community, but they do some investigations, not in a frequent time. From time to time, they do that, and there are not a published paper in the matter.
Okay, because I think this could somehow explain because you don't have cyanobacterium blooms, for example. Maybe the dominance of the species is by mixotrophic species, for example. I don't know if it's a dystrophic lake, you have a very high concentrations of dissolved organic carbon. These probably favor other species other than cyanobacteria, for example. So yes. This could be an explanation. I don't know. You have to go a little bit further in the investigations there.
Yes, maybe we can continue in that line also to just to see all the spectrum. And yes, thank you.
Mariana?
Yes, I will talk something about that, about mixotrophy. And I was curious about your work because you said you have high nutrients, but low biomass, phytoplankton biomass. And I'm not sure if the color of the lake is the humic lakes. Are they humic? In your presentation, I don't remember. But yes. maybe I was going to talk about what Rosemberg said. Maybe it's because mixotrophic species that are there. I'm going to show in my work that with high nutrient but low light availability, biomass decreased a lot, and there isn't cyanobacterial blooms. So I saw in my work that we had blooms of cyanobacteria, but when decreased water level and the water was turbid and light was not enough for cyanobacteria. So even the cyanobacteria survived, and the species that maintained in the system was mixotrophic species adapted to low lights. So maybe it could be that the answer, I don't know.
Yes, that could be an explanation. Yes, we wanted to explain it by the low transparency, but it is connected also. So yes, thank you.
Yeah, there is a lot of work doing about humic lakes and mixotrophy. Maybe this is the answer. If you have the data about phytoplankton composition, it's going to see this tendency.
Yeah, thank you.
Does anybody have any further questions or comments? If not, we should move on to the next talk by Fritz Zingel. His work is entitled as Black and White, Day and Night, Fish Diurnal Movements in Two Different Amazonian Lakes. So,
hello.
Hi, Fritz. Well, does anybody have any questions, comments about Fritz's work? Any questions? I actually have one. You made some comparisons about the fish traits, feeding fish traits in these two lakes, white lakes, and actually we have two lakes, one white lake and one dark lake. And I was just wondering what may be somehow driving these differences in the functional traits in dark and white waters, and why there are some difference in the littoral and pelagic zones concerning the traits, since we are talking about the same system.
Thank you for your question. First, I want to say that this whole study that I'm just presented here is one small part of our bigger topic. Our main interest was actually to find out how and if the methane enters food webs and in these quite shallow lakes, and what are the differences, and therefore we sampled quite a lot of the food web, bacterioplankton, microbial loop components, zooplankton, chironomids, phytoplankton, microphytes, and also did some stable isotope analysis about this. And actually in the parallel session, what is going on now, my co-worker Helena Agasild is presenting about the stable isotope study. And of course, we have quite a lot of data. And to answer now to your question, of course, if we look these different feeding types of fish, there is one reason, of course, is main food object there, and because there are so high species diversity there. There are very many different kind of approach to the prey, and actually it is very hard to put all these when you say omnivores, there are a lot of species behind it, and actually to get the picture more clear, we must divide this group a little bit further, and then we can compare with potential prey objects. And we have also done this, but this is actually too much material to put in this short 15 minutes presentation. And what here is quite, I choose to do quite a descriptive overview about this to give the main frame how this things works there. But yes, it of course the different feeding types in different habitats and in different lakes. Of course, it starts from the fish species composition, and it's also shown it was very different between of course between the dark and the white water. They were very different. We used this Bray-Curtis disseminate analysis, and it was significantly very different. But also we found difference between different habits between littoral and pelagial area. And there were many species that were only in littoral area and many species that were only in pelagial area. And this all leads to these differences in these feeding types. I don't know, did I answer your question?
No, you did. Yes, you did answer my question. Does anybody have any further questions? And thank you for your answer, of course.
Thank you.
I would have a question.
So,
because it is about the diurnal movements of fish. Do you know the mechanism which triggers the fish to move up and down? Do they feel time, or do they go by light, or do they just follow the prey?
I think that there are, again, regarding different fish, there are different mechanisms, but of course the light, I believe, is the main key factor here. But we also found in some species that, for example, regarding planktivorous species, that there were quite nice patterns when they followed the plankton dynamics. And of course, there was also some, we found also some connections to the oxygen patterns. Some fish, but of course there it is sometimes quite hard to take these mechanisms apart. And now it seems that the light is most stronger trigger. But of course, light can affect their prey, and then they can follow the prey.
So thank you for your answer. So there have been no experiments done if the fish also move if you like change the surrounding patterns. I was just wondering because, for example, marine fish seem to have like an inner clock. So if you change the light schedule, or if they travel like half around the globe, then they have jet lag. I was wondering if freshwater fish have the same thing.
Here we found in the dark water lake, we found pattern. We also actually tried to follow, when you put the nets in the littoral area and pelagic area, then you can actually look at from which sides the fish is entered the net. Is it come out from littoral, or is it going from the pelagic area to the shore area? And in some species we found that kind of trend, but not. And again, there are many different species, and when you lump them, then you lose the differences quite easily. And but when you're going to species by species talk, then it takes hours. But yes, some species follow, but it is not the up and down, but it is from shore to the, because it is shallow. It is from the shore to the open, and from the open to the shore in the different times of day.
Does anybody have any further question? Maybe the last one. Well, if not, thank you very much, Prit.
Thank you
for the discussion. And now let's move on to the last.
Rosemberg, you are muted.
Sorry. Sorry, the last talk is entitled as phagotrophy by cryptophyte and chrysophyte across nutrients and light gradients using different measurements techniques. And Mariana is the first author of this work. So does anybody have any questions about this work? Okay, I have. So Mariana, it's just actually a curiosity as well, because you, in your methods, you say that you use beads as prey, and I could not actually follow that. Could you explain a little bit about what is this beads, and why did you use it as a prey in your experiment?
Yes, thank you. First of all, thank you, Rosemberg, for presenting the highlights of my work. And about the beads, it's my work is health ecology and health methodological. And we did some short-term graze experiments, and it's used to in these experiments, in these graze experiments, it's commonly used beads in this to do disease ingestion, to calculate disease ingestion rates. So actually we did some other experiments with fluorescent bacteria, and that it's better because it's a bacteria, it's not a bead, it's a synthetic sphere. So we did this test before with fluorescent bacteria. Algae eats bacteria, this fluorescent bacteria, but we want to see at the flow cytometry. But we didn't saw the signal for fluorescent bacteria in the flow cytometry, so we used beads because the fluorescence is bigger. So we test before with beads with half micrometers, and we also didn't saw in the flow cytometry. And after we used beads with 1 micrometer, and we were able to saw in the flow cytometry. That's why we use it. So it's commonly used beads, and we use these beads because we want to saw in the flow cytometry. It's a different technique that we were testing, a new protocol for estimating these grazing rates for mixotrophic species.
Okay, thank you for your answer. Does anybody have any further questions? I have actually a final question about, my last question about your work is, I was wondering, I was trying to find a hypothesis for your work, and I could not find it. And also was thinking about the relevance of your work concerning this mixotrophic world, let's say, because I could not find it in your presentation and also in your abstract. If you could say something about this, would be nice.
Yeah, I'm sorry for that. Yeah, I have a hypothesis that actually I have two, because I did two kind of experiments. One short-term experiment to see grazing rates of two kinds of mixotrophic species, one cryptophyte and another chrysophyte. And we did in different levels of light and different levels of nutrients. So we want to see how ingestion rates change through these levels of resource for phytoplankton. So we expected to see that with with low light and low nutrient, mixotrophy will be, grazing rates will be greater, will be higher. But we saw that there were difference between the two species, and cryptophytes had the higher grazing rates in high nutrients. And as I was told in Claudia, was expected because we want to answer a pattern that we saw in the nature. So we saw in eutrophic systems, cryptophytes were there, but light was limiting. And we want we expect to see this in high nutrient, but in low light or dark, and this species will have have higher grazing rates. So, and we expect for the opposite for chromonas, that chrysophytes, that in the literature, in low light and dark systems, will have higher grazing rates. It's what we found. And the second hypothesis was for the second experiment, because we want to see, we use a different technique with Lysotracha to mark food vacuoles in the species. And we want to, first of all, see if with this methodology, we can separate, we can distinguish autotrophy from heterotrophy, and we saw that. And after, we want to see if it's possible to quantify this difference. And we didn't manage that. We didn't solve that. So we only see that Lysotracha is a good tool to see qualitative. So we can distinguish mixotrophy and autotrophy and heterotrophy, but there wasn't difference in through the treatment. So because we expect to see that more Lysotracha, more mixotrophy, but it was not possible. And the relevance, the second answer of the second question. And the relevance through the field is because when we study mixotrophy, a lot of work is on oligotrophic systems, is on oceans, and little bit is known in eutrophic systems. And here the most systems we have are eutrophic, and we need much more effort to calculate these grazing rates, to much more understanding. So it's in this sense.
Okay, thank you very much for your answer, Mariana. We have time for a last question if anybody wishes. That's the time. Okay, if nobody has any questions, I think we can end our session. I'd like to thank you all for your attention, and I think we had some very interesting discussions here, and well, I hope you enjoy the last 3 days of the conference as well. We have this session is going to take place again on Friday, and well, thank you very much for everything.
Thank you to you, Rosenberg.
Thank you.
Thank you for organizing. Bye-bye.